All right. Hi, everyone. So welcome to the first webinar of the NAPB Graduate Student Working Group for 2024. And we are all excited for today's talk. So by the way, just a couple of house rules. So we have a designated question and answer after the presentation. However, if you have questions during the presentation, feel free to chat them in the chat box, and Dr. Jim will be happy to address them along the presentation as well. So this webinar is being recorded, and it will be also posted in our NAPB Graduate Student Working Group YouTube page. So if there are things that you would want to revisit, or if you would want to watch the webinar again or share it with your friends and colleagues, that will be really awesome. So with that, we will start the webinar, and Andy will introduce our today's awesome speaker.
Great. Dr. Jim Holland received his bachelor's in biology from Johns Hopkins University, a master's in plant breeding and genetics from the University of Wisconsin, and a PhD in crop science from North Carolina State. He was an assistant professor at Iowa State University for 5 years and moved to the USDA Agricultural Research Service, where he's at now in Raleigh, North Carolina. And I give it to you, Dr. Holland.
Okay. Are we looking good now? Let's see. I gotta switch it again, don't I?
Yep.
Got it. Okay. All right. Thanks very much for inviting me. It's a nice opportunity. We'll talk in general terms about the population structure and diversity of maize and what the implications of that have for breeding and long-term corn improvement. So the story of maize starts about 9,000 years ago in southwestern Mexico. Before that time, there was no maize. There were wild grasses called teosinte, which don't really look much like maize. The tassels here at the tips, they do look like maize tassels, but most of the rest of the plant doesn't look very similar to maize. In fact, the ears look completely different than a typical maize ear. Even a primitive maize ear looks very different from a teosinte ear. But humans domesticated this species and essentially turned it into maize. So maize is a human creation, and they did this by selecting for, in part, some major mutations that do things like reduce the dispersal of the seeds. Instead of the seeds shattering at maturity, they hold on to the cob. There's a single major gene that reduces this hard seed coat so that the naked grain is exposed and it's easier to eat. And the branching patterns of the plant were also changed by a single major mutation that humans selected for. So this was probably the first major event, obviously, in the population genetics of maize was the domestication event. And if you look at genetic markers throughout the genome, and you create a dendrogram of the relationships, there's a very clear split between the closest wild relative of maize, which is this teosinte subspecies parviglumis, and all the maize types down here. So when people look at how much diversity is lost at SNP loci, maize has about 83% of the diversity that teosinte has. So there's, right off the bat, there's this loss of diversity. Now this bottleneck due to domestication is not so severe like crops where there's polyploidy events or things like that. The bottlenecks have been much more severe. So it's not quite so bad in maize. And part of the process is this domestication occurred over probably 1,000 or so generations, something like that. And also because there was gene flow after the domestication event. So even after you had something that looked like maize, there was still some crossing back and forth between teosintes and maize. This has been highlighted in a brand new paper that just came out like a month ago in Science, where if they look at a lot of germplasm and a lot of teosinte samples and a lot of whole genome sequences, basically, what they've shown is that there was a divergence between two subspecies of teosinte, this parviglumis, this closest ancestor. This is a truly wild form. It likes to grow in undisturbed areas in Mexico. And then there's a subspecies mexicana, which is weedy, and it likes to grow in disturbed areas. And now it turns out that there's pretty good evidence that there was gene flow from this other subspecies mexicana that came back into maize after the initial domestication event. So the only maize that's ever been found that doesn't have evidence for this mexicana introgression is this ancient cob found in Peru from about 5,000 years ago. So everything else, so there was sort of an initial event that only involved parviglumis. There was a primitive maize, but at some point that primitive maize had introgression from mexicana, and those things have intermated with each other and with ancient mazes to form what we have now. So if you look at the archaeological evidence, again, the initial domestications here in southwestern Mexico about 9,000 years ago, and then maize spreads very quickly out of that. Well, you know, 1,000, 2,000 years, it's already moving into South America. It takes a little bit longer for it to move north. One of the things you'll notice why it could move south quicker than it could move north is because this is a tropical region here, and as it moves north, it's moving into subtropical and then temperate regions where the day lengths are different during the growing season. But all this movement and spread of maize occurs before Europeans ever arrive in the Americas. So what's happening in this process is that there's a combination of natural selection to these different environments along with human selection. So this is a little map of the ecological zones in Mexico. So just looking at Mexico, there's a huge variety of ecologies, and the maize that's grown in different regions of Mexico has adaptations to these different environmental factors. So that's basically, if maize can't produce seed under these different environments, there's natural selection that's going to take it out. But there's also an element of human selection where the people in these regions are selecting maize for the different kinds they want. So you end up with this massive variation in the land races of maize. So these land races are managed as open pollinated populations, and farmers have maintained them for literally thousands of years. And not just maintain them, the farmers, these are actively evolving populations. The farmers are still selecting out of their fields the plants and the ears that they want for various purposes. They are saving seed for future plantings. And the farmers are actually pretty knowledgeable about how to maintain different varieties in the same region and keep them separate. And so things like sweet corn, popcorn, flowery corns, flint corns, dent corns, these were all developed by the indigenous people of the Americas before Europeans arrived. So there is this really key element of not just ecological adaptation, there's a really big element of human selection for the kinds of maize they want to eat, ceremonial purposes, and things like that. So the domestication is the first bottleneck. The next one, as I pointed out, it took a while for maize to get up into the temperate zones. And actually this involved another bottleneck that can be observed both at some of the key genes involved in photoperiod sensitivity. And this is just a map of the different alleles at one of the key genes. And you can see it's not complete exclusion of the original tropical alleles. You still find some of these things up here, but these red dots are basically novel, later appearing variants that reduce the photoperiod sensitivity. You can see the frequencies are much, much higher in the high and the low latitude zones of the Americas. So if you look at a collection of inbreds from all around the world and just run a bunch of marker assays on them, you can see that, again, there's clear differentiation between sweet corn and popcorn. But even within the dent corns, there's a big differentiation between tropical inbred lines and the temperate inbred lines, which here we've subdivided into non-stiff stock, and stiff stock is the major breeding groups of the US. So it's clear that if you want to get novel alleles, you can go into tropical inbred lines and identify, or at least obtain, some kinds of different genetics by breeding. But part of this differentiation between temperate and and tropical kind of gets exacerbated by the development of the inbred hybrid breeding method. So this was actually proposed by Jones. This is 100 years ago now, where he showed that, yes, if you make inbred lines, they get short, they're weak, they're not very good, but if you cross unrelated inbred lines, you can make F1 hybrids that are quite robust. So here's a picture of the famous B73 inbred from Iowa State, Missouri 17 from the University of Missouri. If you cross these things, you get a very robust and high-yielding hybrid. So what breeders have done, basically for the last 80 to 100 years is move from populations to developing inbreds. And then once they had inbreds that were pretty good, those things were intermated to each other. And so there's this recycling process where if you look at the pedigrees of commercial inbred lines that were developed by companies, that they will trace back to these important ancestral lines like B73 that came out of the public sector, but they then get recycled kind of over and over within the commercial companies. So there's really a strong reliance on a relatively small number of inbred lines in the pedigrees of modern hybrids, and this closure of these pedigrees means that there's very little sort of new genetics coming into the system. And if you ask the question, how much exotic germplasm is there in commercial hybrids? It's very little, and what exotic germplasm there is typically something like European exotic. It's basically temperate germplasm from another region. It's very little of it's tropical in origin. And SSR surveys of allelic diversity have shown that within the commercial companies that the diversity has dropped. So, okay, diversity is going down as a consequence of this recycling of inbred lines in commercial breeding programs. But on the other hand, if you look at the average yields of maize in the US, basically since the adoption of this inbred hybrid breeding method, yields have been going up at a fairly astounding rate. So one might then just ask the question, well, okay, so what if there's a loss of diversity? So what if there's bottlenecks? These are a good thing. I mean, in fact, this is what we do as breeders. Selection is supposed to be eliminating bad alleles and enriching favorable alleles. And that's what seems to be happening. So that should result in a loss of diversity. So to some extent, this loss of diversity is actually what we want to make better crops. So this is kind of like a fundamental thing that happens in breeding. So if you're a successful breeder, you're probably going to see some loss of diversity. But on the other hand, there's this sort of, in the back of your mind, you have to ask the question, that idea is kind of a perfect idea if at the beginning in your breeding pools, you already had all the favorable alleles. And then all you're doing is increasing their allele frequencies. But you can ask the question, did we start with all the favorable alleles in the Corn Belt dent maizes? And I'm not going to answer that directly here, but I'll show you an experiment where we asked an even more basic question, which was, did we capture all the good alleles from teosinte, or does teosinte have some alleles that actually are better than some of the things that made it through the domestication bottleneck? So this is an experiment where we sampled teosinte and a maize landrace population from basically the same town in Mexico, and we evaluated them in field experiments for a whole suite of traits, and they're very different. For any trait you measure, these subspecies are very, very different, as you'd expect. Maize really looks quite different than teosinte. But if you measure the total seed weight per plant, which is something like yield, individual plant yield, they don't differ at all. So the domestication process, and this is a land race that's sampled probably in the 1990s. So it went through domestication bottleneck and subsequent selection by humans for thousands of years. It's no better in terms of seed production than the wild relative it came from. For me, I found this to be a somewhat shocking result. I assumed that for sure the crop species would be, have higher yield potential than its wild domesticate. But that's not true. So then the question is, what were humans selecting for over these thousands of years? And mostly it's ease of harvest and for, how these things taste, ease of turning this thing into food. And those are all really key traits for domestication and post-domestication, but they don't necessarily increase seed yield. So, maybe it shouldn't have been that surprising. The other thing we did in this experiment was we compared outbred versus inbred, single generation of selfing within each population as a way to measure how much quote unquote genetic load there is in these different populations. So by selfing and increasing the homozygosity, if you have deleterious alleles that are unfavorable, particularly when they're homozygous, but maybe masked when they're heterozygous, and so you don't necessarily see their effects on outcross populations. The question is, how much inbreeding is there in maize versus teosinte? And this kind of shocked me as well, is that maize actually has substantially higher inbreeding depression than teosinte. So for example, in this graph over here, this is the total grain weight per plant. This is kind of our proxy for yield on a single plant basis. Teosinte definitely has inbreeding depression. It's about 27% reduction by a single generation of selfing. But in maize, there's a 50% reduction. So there's actually a higher genetic load in the domesticated species, despite these thousands of years of human selection. And if you look at the sequence data, there's ways to sort of predict what genetic variants solely based on sequence information might be deleterious. It turns out that even just looking at sequence data, you can find in these landrace populations that they're segregating for a higher predicted genetic load on the sequence data. And it turns out phenotypically that's true. The story's a little bit subtle. If you look for large effect variants, like kind of almost Mendelian segregating things that are either highly lethal or have really large effects, there are more of those in teosinte than in maize. So the domestication bottleneck actually knocked out some of the large effect deleterious variants. So that's good. The bad news is the polygenic load, the kind of all the bad alleles that are recessive but have very small effects, and there are many, many, many more of them, cumulatively, their effects are greater in maize than in teosinte because the bottleneck allowed drift to become more important and actually reduce the effectiveness of selection. And so you have this higher polygenic load. And the really bad news is polygenic load is harder to breed out. It's not just like a single gene or two genes where you just get rid of them. It's all throughout the genome. So that's the bad news. So that's sort of an academic way of looking at this and saying, well, there's probably better alleles even for like yield in teosinte, but it's very hard to just go into a wild relative and extract favorable alleles because they're linked to all these other things you don't want. So it's probably easier to work with things like land races or just more diverse inbred relatives. But there's evidence, if you don't look at yield, you look at things like disease resistance. So here's an anthracnose resistance gene that was identified only in tropical lines and is actually now deployed in commercial hybrids from Corteva. There are, CIMMYT's discovered very low frequency in land races, high levels of resistance to tar spot, which is a relatively new disease that's particularly problematic in some tropical regions, but also is in the US now. If you go all the way back to teosinte, here's a group from China, Feng Tian's group, that has identified genes that modify the plant architecture that actually can increase yield under higher densities. So that's just from maize. It's been kind of difficult to actually show this directly in maize. If you look at other species, particularly the selfing species, there's plenty of evidence that shows that those domestication bottlenecks did in fact leave behind lots of useful genes that you can integrate into your modern crops. So we would like to do this. We would like to go into this more diverse genetics, maybe into teosinte, although that's pretty hard, but at least into like tropical maize and try to kind of leverage that genetic diversity for our breeding programs. But it turns out it's difficult. One of the big problems is just simply the photoperiod sensitivity. So tropical maize is highly sensitive to long day lengths. It basically will just continue to grow vegetatively until it gets a short day exposure. So here's an example of this is a commercial hybrid that's already flowered, grown in our environments in North Carolina, next to a population of Tuxepeño, which is a landrace from Mexico. And there's a few tassels in there, but there's basically no ears. And most of the plants haven't even put out a tassel yet. It just keeps growing taller and taller vegetatively. So we would like to access this kind of material for our breeding programs, but it's totally unadapted and you can't just work with it directly. So one thing you could consider is, what if we studied the genetics of photoperiod sensitivity, identified the genes involved, and then we could just kind of breed those things out if we know where to go. And there's evidence, again, from other crops, and here's an example from sorghum, where there's like really large effect single genes that if you can identify those things, you can sort of quickly breed them out and do fairly rapid adaptation. Unfortunately, it turns out that things that are simple in selfing species and species like sorghum are often much more complicated or much more polygenic in maize. So you can go back to 1967, and even then it was so clear that there were large effect variants for photoperiod sensitivity in sorghum that they had named genes. You know, they didn't have markers, they didn't have sequence data, but they knew they had major genes that could be mapped with classical linkage mapping. And if you compare this MA1, maturity 1, the difference between the homozygous classes at just this single gene causes a 34-day change in flowering time. So it's a massive effect from a single gene. And so sorghum's been very effective. Sorghum breeders have very effectively broadened their genetic base by crossing to, you know, African sources of germplasm that are photoperiod sensitive and just quickly kind of breeding these major genes out. The problem is in maize, when we've mapped the photoperiod sensitivity genes or the flowering time genes, the largest effect allele we've ever found in maize is a 3-day effect. So it's 10 times less than what you'd find in sorghum. So this is very sad. It just means it's going to be harder in maize. Here's basically the direct evidence for that. So we have this nested association mapping population where you have a bunch of biparental families developed from crosses between these parental lines here and B73. We measured the photoperiod sensitivity of all the lines from these crosses, and then we mapped a series of QTL. So this heat map is like along the rows, these are the QTL. So this is our large effect, this 3-day effect variant on chromosome 10. And you see the heat map. So a few tropical lines have very strong, for us, very strong alleles. That's a 3-day effect. But actually, most of the tropical lines don't even carry an effective allele there. And then the effect sizes decrease. So these are QTL. You can pick them out. We can map them. Actually, the underlying genes have now been cloned in these major QTL cases. But their effects are typically on the order of a day, something like that. So they're not huge. And our top 4 QTL explain only about 25% of the variation. And everything after that is kind of goes from oligogenic to really polygenic. So lots of genes with relatively small effects. This has implications for modifying flowering time in maize using transgenes and gene editing, that if the genetic effects are so small, maybe this is not a trait that's a good target for editing. I'll say that, and then I'll say, nevertheless, we're trying anyway. It's possible that something like a gene edit at a gene that the natural variation is not very big, maybe by an edit, maybe a full knockout is going to have some bigger effect that maybe is more effective. Or maybe the knockout messes up too many other processes and then it's not going to work. But anyway, we're going to try. So the question is, how do we overcome these bottlenecks and reintroduce useful diversity in the face of this polygenic inheritance? So just to recap what I've said, You could back cross, you can use back crossing programs, but if it's mostly polygenic, back crossing only affects a small proportion of the genome, so it's not likely to have like huge effects. And again, despite the fact that this is probably not going to work, we try these things anyway. So there are probably special cases where sometimes this might work. You can do the other way. This is again what the sorghum conversion program did, was they would basically introgress adapted like photoperiod insensitivity alleles into tropical germplasm to get them quickly adapted. But again, our genetic effects are, even at the major adaptation genes, are not big enough to expect that this is going to be highly effective. And again, we're going to try anyway. We're going to do gene editing to see if that's a quicker way to do this. We're also doing actually something like the sorghum conversion program, where we're trying to back cross the insensitive alleles at major photoperiod genes. But I don't think it's going to be a big enough effect to be generally useful. So here's sort of a list of things that I think we're either working on it now, or we have evidence, or other people have evidence that these things actually can be reasonably effective in maize. And number one is kind of obvious. I'll show you, we have a lot of germplasm collections. Sometimes the problem is even knowing where to start, what's the good material. So we need to evaluate the germplasm collections in our own environments to see what's going to be useful. And then if you have things that are, for example, tropical breeding populations that maybe are going to be useful, but they're not adapted, well, the first thing we really need to do is adapt them. So are there ways that we can use kind of pre-breeding methods to overcome the adaptation barrier, so that at least we can get them into a phenotypic shape to use in crosses with our elite material? And then going from populations to inbreds, as I showed you, there is a collection of tropical inbreds, and there's programs like CIMMYD and IITA that are public programs generating tropical inbreds. Is there a way to use those things effectively in temperate environments? And Major Goodman, who recently retired from NC State, did this for many years, and I'll show you some of his success stories. And then the last example I'll give is maybe the one that's sort of closest to having impact on commercial breeding programs, and that's the germplasm enhancement of maize, where you take exotic material and directly cross it with elite commercial germplasm and select for favorable progenies from those. So germplasm collections are really important in maize, because, as I said, and many crops, because as breeders have been successful in deploying improved cultivars, the old landrace varieties basically got abandoned and would have been lost and extinct if they hadn't been collected. Now, and some of them did go extinct, but many of them were collected, particularly in maize, during the time where hybrids were being adopted by farmers. So CIMMYD and the USDA both have really extensive collections of maize germplasm, which includes populations, includes inbred lines, both of those things. And if we look at the inbred lines are nice to work with, because it's much easier to do sequencing on them, because you only need one plant, for example, from an inbred line, and it's mostly homozygous, and it's easy to deal with. Again, I already showed you this picture, but if you look at the inbred lines of the USDA PI collection, you can clearly discriminate most of the tropical lines from the temperate lines. And so this gives you a way to at least identify that, yes, we're working with things that are genetically distinct. The difficulty here is that many of the collections are open pollinated varieties, and that's much harder to deal with in terms of sequencing, because within a collection, there's lots of genetic variation, so you have to sample more things. So this is something we're trying to grapple with right now, and dealing only with the open pollinated varieties that were collected in the USA. So the advantage there is that in many cases, we don't have the adaptation barriers to worry about, but we do have the issues of, like, genetic load is big. They haven't really been bred for, ability to produce inbreds, or, lodging resistance or things like that. But we suspect that there's probably some useful material in there that's really relatively uncharacterized. So over the next 2 summers, with support from a NEFA grant, Sherry Flint Garcia and Jacob Washburn, who were with USDA at Missouri, and their graduate student, Melissa Draves. They're going to evaluate materials there for 2 summers. And here with Joe Gage at NC State and Jordan, graduate student at NC State, we're going to evaluate all 1,000, pretty much 1,000 of these collections in the field. And then we're going to try to do at least a subset of them. We're going to do some sequencing analysis on to better understand the trait diversity and the relationships. And again, part of this is aimed at with this goal of like, maybe there's material here that's going to be useful for like corn belt dent commercial germplasm. But there's another goal, which is that this material in and of itself has a lot of interest, maybe not for commercial breeding companies, but for people who are interested in specific food uses of corn. So in the South, grits are a big thing, but tortilla production, things like that. It would be, there's a lot of people really interested in producing better quality tortillas, and there's a huge amount of variation in this material that some things are going to make better tortillas than others, for example. There's people who are interested in distilling, and there's also a cultural importance to these things, where this is a group of people from the Wakamasiwan tribe, which is an indigenous group in North Carolina, and they are rematriating some of these varieties for cultural purposes, because these are things that their ancestors grew for probably thousands of years, and then some of that culture was lost, and they'd like to reclaim that. Okay, so that was one, is like, yes, evaluate the germplasm collection, see what's in there. Step 2 is like, if you have populations that are interesting, but they're unadapted, we need to select for adaptation. So this is an example of a Caribbean tropical landrace population called Tucson, and Arnold Hollauer at Iowa State tried this, where he took the original population, grew it in Iowa, and then for 10 generations just selected for the earlier flowering plants. And the phenotypic difference due to this selection is pretty massive. So this is the original generation 0 population. The mean flowering time was something like 82 days after planting. And then by 10 generations of selection, Arnold Hollauer was able to reduce that by about 20 days. So this is not adapted, but this is basically in the adapted range. So he, just by doing phenotypic selection and repeating recurrent selection, you can move the phenotypes of these populations. We had the question of like, what's the genetic basis of these differences? So Randy Weiser and another group of colleagues, we looked at this, and we basically just looked at SNPs throughout the genome, and we asked the question, like, how much allele frequency change was there at these SNPs? And so that's what's plotted on the y-axis. And then how much effect can we measure them due to these genes using like a genome-wide association study. And that's here on the x-axis. And you can see there's a correlation. So the bigger the phenotypic effect of the SNP, basically the faster it was selected against. This is this chromosome 10 major gene there, and it pops out as, again, having the biggest effect in this study. Now, its allele frequency change is not so big, and that's because it started at relatively low frequency, and within probably 3 or 4 generations was completely fixed. So all the highly sensitive variants, because they're big effect, they got flushed out almost immediately. So that was sort of the first phase of the selection process. You can see like there are major gene effects that are like, boom, they're gone from selection. But after that, there's another 6 generations of selection where you can see the response keeps going and going and going, almost at the same rate. And that's based on polygenic effects. So we identified about 16% of the genome shows some signal of response to selection. And so the story here is, yes, there's a few sort of major genes, but there's a lot of polygenic stuff that needs to be selected on if you're going to really change the population phenotypes. Here's the good news. If you do this adaptation process, then you basically improve not just flowering time, but there's a lot of other traits that depend on reasonable flowering time for adaptation. And this is kind of an obvious point, but we showed this quite directly here, that we compared this is a different population, but we compared populations that showed more or less response to selection. So on the x-axis, the farther they are to the left, they've reduced flowering time. And so that's what we want, because they flowered too late. And then if you look at things like plant height comes down. That's a good thing. Tassel weight comes down. The really original material has these gigantic tassels, which are kind of unnecessary. They get smaller. And kernel number and kernel weight go the other way. So as you decrease flowering time, you actually get more kernels per plant, and you get higher seed weight. So that's great. This is the kind of like, this is just showing that, yes, you have to overcome the adaptation first before you can even get things that'll kind of produce reasonable yields. And then just very quickly, part of this experiment was to demonstrate the effect of doing this kind of adaptational selection in different environments. So we had 8 different environments at different latitudes where we conducted the selection. And these were all like independent selections out of the same base population. We evaluated them in the same 8 environments. So for example, this column here is we selected in Puerto Rico, and then looking across the rows, here's the population we selected in Puerto Rico and evaluated in Iowa. And the heat map shows there's not much response to selection for flowering time. If you select in Iowa and evaluate in Iowa, you get this very strong response here. Obviously, it's direct selection. But this is a little bit problematic because it shows that if you do this selection under these short days, you're not exposing the photoperiod response. You can't really use these environments for adaptational selection. And the problem there is that cuts out half of your potential generations per year. So it's going to slow down your selection response. So we'd like to be able to use off-season nurseries for adaptational selection, but just the direct phenotypic selection there doesn't look like it's very effective. So we did a follow-up study where we said, okay, let's train a genomic selection model in this population in our summer temperate environment, and then use that information to genetically evaluate progenies in the off-season nursery under the short day lengths, and then see if we can predict the flowering time of their offspring in the next generation. And it turns out it's not perfect. It's not, maybe not even great, but actually it's good enough. This is the correlation between what we observed in the next summer after doing the off-season genetic predictions versus the predictions here on the y-axis. The correlations are like 0.36, 0.35. So they're not super high, but actually this is limited due to the heritability of the training set, because we trained these data on individual plants in our summer nursery. So that kind of limits how effective you're going to be. But still, even with this level of correlation, we figured you're going to be more effective using genomic selection in the off-season nursery than direct phenotypic selection. And you can almost recover, not quite, but you can almost recover that sort of second effective generation per year. And if you could optimize this to probably do better and maybe do multiple off-season cycles of selection. Okay, so just a couple more things. And so that's like population-based breeding, that genomic selection could be useful there in those sorts of populations. Here's an example of just pure phenotypic selection done by Major Goodman over several decades here in North Carolina. And basically what he did was, early on, he sort of realized, you know, land races are pretty difficult to work with, and they're not going to generate good inbred lines very quickly. So what he did was he really concentrated on using hybrids or inbreds from tropical breeding programs. These have already been purged of strongly deleterious alleles. You can get inbreds out of them much more easily. The big issue is overcoming adaptation. And it turns out you can take 2 different tropical lines, as long as they're not too closely related, cross them together, and you'll actually generate a lot of variation for things like flowering time. You can actually just select for earlier flowering in those kinds of crosses without ever crossing it to adapted material. And you can essentially adapt these kinds of materials. Takes some time, but it can be done. The beauty is when you get an inbred line out of this process, you know, after his selections, he identified a number of these that had quite high heterosis when you cross them to a temperate inbred line tester. So Major, you know, he's not one to ever blow his horn. He would always just say, "Oh, I just produced a bunch of trash." But actually, his material was it's quite impressive how this was done with almost shoestring budget, but really just keeping focus on it over many, many years. And so if you look at the genetics of this material, these are principal components plots, just to show you that here's like typical corn belt dent materials. You know, this is a typical hybrid, is a cross between these groups. And Dr. Goodman's material, these temperate adapted all tropical lines, really are quite distinct. You know, doesn't really matter which principal component you look at. They're really outlier groups, and they also have good yield potential. So if you evaluate the yield of these things compared to, these are crosses between kind of old, a little bit older, but these are commercial line F1 hybrids, these two things here. If you take one of these F1 hybrids and you cross it to one of his all tropical lines, he actually gets higher yield in the three-way cross. You shouldn't be beating an F1 with a three-way cross, but he does. And the other thing is, it turns out you can cross this tropical line to either stiff stock or non-stiff stock, and you'll get this boost of heterosis. So it's really kind of a distinct heterotic group that's been developed for use in the US. Now, the last little point here is that industry, you know, the commercial breeding programs are sort of like, yeah, this is all sort of interesting, but we need a new product like coming out in a few years. We're not that interested in these kind of long-term things. So the USDA germplasm enhancement and maize program was developed to basically institutionalize a little bit of what Dr. Goodman was doing, but make it more applicable to you know, the dent corn seed industry. So going back to this idea where the whole process needs to start with evaluating germplasm collections, there was a multi-year, very large project called the Latin American Maize Project that involved most of the countries in the Americas, where they evaluated their own germplasm collections and identified basically landrace successions that had really good yield potential in their own environments. So accessions that were identified in this program as like having potential good agronomic performance, but they're totally unadapted. Those then were chosen by the Germplasm Enhancement of Maize program to be crossed to elite inbred lines from companies. And actually the companies make these crosses. So they'll make this cross, generate an F1, and then they'll turn over the progenies of that F1 to the GEM coordinator. So Matt Krakowski runs the GEM program here, located in North Carolina. Adam Vanus is the GEM breeder at Iowa State. Iowa State, because they're farther north, they need another back cross to the private company lines to overcome the adaptation barriers. But basically then it goes into straight phenotypic selection among lines, and then they make hybrids, they test these hybrids, and the germplasm gets released. So the key thing here is that if you look at, so this, again, there's always these two questions. Are these lines really genetically different? And are they any good? So in terms of genetically different, this is again another principal components plot. The temperate parents of these crosses are kind of like these red dots and these blue dots, which are kind of in this region. And then if you look at the GEM lines, either the parents or the progenies of these crosses, they kind of really push out the variation in kind of in new directions. So they do represent new diversity. These are the box plots of yields for a bunch of different crosses. So each of these is a biparental family. But in this case, they're 25% tropical, crossed to a temperate tester, evaluated in a bunch of environments. And here's commercial checks. So like some of these families are doing as well as the commercial checks. Most of them are not. That's okay. That's kind of normal. But even in the crosses where the mean is not as good, you'll see there are some outlier progeny in the favorable direction. So there's like good material coming out of this program. So we see there is a question. I could stop here. I'm actually pretty much done. So I guess I'll just keep going. This is the summary points. These are lab mottos that I tell my students and the people who work with me, and they think I'm kidding. And then after a few years, they realize, oh my God, he's right. And then they say them. So one lab motto is that selection works. Like constantly amazed that, like, if you have heritable variation and you select and you do this for several generations, you will get selection response. So breeding works. It's really highly effective. Again, that should not be a stunning conclusion, but I'm constantly sort of, wow, that's cool, it works. The corollary or the counter motto to that is like, when you start something, you think it's good. Wow, it sounds good. You think it's going to be easy. Maybe you read a publication, somebody proposes this method's going to be great. It always sounds like it's going to be easy. And then it turns out to be a real slog most of the time. So all these things I'm proposing take a lot of time, a lot of effort. There's no real trick to this. Like, you just have to try this method, or there's some new technology that just suddenly makes everything easier. Each little advance in terms of technology and breeding, it's like polygenic. It adds a little bit. It gets better and better, like a little bit at a time. But you have to keep combining all these new approaches and kind of keeping the selection pressure on, and do this over time and generations to really make progress. And then the last thing is that I wasted 45 minutes of your life telling you what I know about corn breeding, but the reality is I can't teach you how to breed corn. I don't really know how to breed corn. I can teach you how not to do it because I've done many things wrong, and I could give probably a more effective seminar on all the stupid things I've done that didn't work. So you can ask me about that sometime. I can tell you what not to do. And again, the things that actually worked was because there were lots of good people involved, lots of collaborators at many institutions. So I don't even have time to thank everybody, but I want to throw some names up there and show some pictures of the various crews of people who've worked with me over the years. This is my current group now, and you'll see that people are working on, again, we're trying lots of things because we know some things aren't going to work, or maybe the way forward is trying lots of different things and like figuring out how to combine ideas. But for example, Destiny's trying to do gene editing of these photoperiod genes. Stefano is actually doing the back crossing. So we're kind of like comparing how effective these different methods are. We have people doing things like the Circa project, which I'm not going to talk about. You may hear about it someday. Not time to do it. Jordan's working on the land races. Anna and Nicole were here, and then they did good stuff, and then they got their degrees and good jobs elsewhere. And Chuyer's working on genomes to fields and domestication work. Eric, who right now is sort of on a mini sabbatical with CIMMYT in Zimbabwe, he's working on disease resistance and also the adaptational selection program. And Francois, Sambao, and Josie are like making sure everything works. So I will, last little propaganda advertisement is that there's a corn breeding research meeting hosted here in Raleigh at the end of February. It's going to be awesome. You should come. Even if you don't work in corn, it's going to be so good. You can convince your supervisor that you should come to this. Okay, I'm done. I'll take any questions.
Okay, so yeah, thank you so much, Dr. Jim, for the insightful presentations. So I think you have covered most of the questions during webinar, but even we have many questions like that student posted during the registrations. The first one have what could be the best approach to introduce genetic diversity in a breeding program?
Yeah, right. Yeah, so I would go back to my little work. Let me before I go too far, let me just clarify that what I have said here, I think is maize specific. Some of it. It's not completely general, because I think, for example, if you're working in wheat or soybeans or rice, the story is kind of different. First of all, I think in those species, there are many good examples of large effect genetic variants, even from wild relatives, that can be introgressed and have very important effects in the context of the crops themselves. So, I think that's what makes it more challenging in maize, is that just introgression alone may not always work as effectively as you might think based on how well it's worked in other species. But I think even talking about wheat or soybeans or anything else, you have to start with some knowledge of the germplasm. How different are the germplasm resources you have available? Is there any reason to go into them? Do they have trait variants or trait effects that you're really interested in? And the challenge there becomes in, how do you know if these germplasm resources carry genes for traits that you're interested in, or favorable genes, if the adaptation problems are so severe you can't even measure the traits? And so that's the problem we have in maize and in probably other species, where if the thing doesn't flower in your environment, how do you know if it has any potential yield in your environment? So you have to overcome those barriers first. So if you have large effect variants that you've identified in your germplasm collections, backcross away. I mean, I think that clearly is the efficient way to do it and can be very effective. But if it's polygenic, I think we just have to face the facts that it's going to take long-term kind of pre-breeding and kind of germplasm development before you can get to making crosses with your elite material.
We have another one. Is it effective to use virus-induced gene silencing to silence a gene in polygenic trait? Because in that case, we know that genes are all over the genome.
Yeah, what I'm suggesting is it's if the trait control is mostly polygenic, if you don't have a major effect variant or a major effect gene, then the silencing can work. Like the technology can work exactly as stated. Like it can silence some gene, and maybe that silencing has some favorable effect, but it may be that the effect is very, very small. And so the issue then becomes with any of these new technologies is the effort and cost required to do this kind of alternative genetic modification, even if effective, does it have enough kind of phenotypic impact for it to have been worth it? And you have to kind of evaluate that on a case-by-case basis. Most traits have some kind of spectrum of genetic control. That often there is one or a few larger effect genetic variants. So there may be cases where if you hit the largest effect gene and do something like a silencing or a knockout, maybe that's going to be useful. I will also say that for many traits, knockouts and silencing maybe are not what you want to do, right? You probably want some perhaps subtle modification in gene expression, whereas some of our tools are still, even though very advanced in some sense, they don't really often give you the ability to kind of fine-tune gene expression. They're typically kind of brute force knockouts, and maybe that's not always what you want. So there will be a few cases where these things are effective. They're going to be more effective when you have large effect genes that impact the traits you're interested in. And they'll be, as the corollary, they'll be less effective the more polygenic your trait is.
The another one exciting one is what is the recent alternative breeding techniques to counter bottlenecks of using heirloom maize varieties?
Right. So, I mean, well, first of all, there's old school stuff still works. I mean, it's kind of the selection works is really true. So, in terms of breeding methods, one thing we're thinking is if there's a particular heirloom variety that has like a particular niche use, let's say it's really good for some food quality, we may be able to improve that variety just by within variety selection. So we're not necessarily going to cross it to anything else. We can do something like recurrent S1 selection or something like that. And actually, we've started doing that. And again, it seems relatively effective. That's nothing new. In terms of like alternative technologies, I think the thing that would come to mind again would be something like genomic selection. Probably going to be most effective if you're doing that within, you're training and you're selecting within that single variety or single population. I think genomic selection is going to get a little bit hairy as soon as you start crossing out of that the germplasm group you're most interested in improving, if you cross to elite material, for example, and then evaluate yield potential, I think your genomic selection model is just going to drive you back toward the elite material. So that's kind of why I think you have to be a little bit careful with things like genomic selection, that we want to kind of, for the pre-breeding stage, keep that separate from the crosses to the elite material until your population's improved enough. In terms of like GM technologies and gene editing, I mean, I don't know what trait I would even target for those things. There's also an issue of we're kind of targeting niche grower and producer groups who are probably more interested in conventionally developed materials. So I don't really, right now, I don't really have a specific vision for, kind of genetic modification for these heirloom varieties.
Yeah. Okay. I have the last one, like we are running of time, so then I'll turn over to you, John. So the last one is like, what are the key challenges in balancing the selections for desirable traits, like such as yield traits in maize, with the need to maintain genetic diversity?
Yeah, the answer to that question depends kind of on what your objectives are and who your employer is. So I have a very different balance than a commercial corn breeder does. So a commercial corn breeder, I mean, I've talked to them and they're really not worried about diversity. They say we kind of have the diversity we need. You can see the yield gains are still going. And as long as that happens and there's no like new emergent disease to worry about, they're going to just keep riding the elite by elite type crosses. And that makes sense. I mean, that's where you're much, a million times more likely of getting a new variety of an elite by elite cross than you are from going into something kind of like a land race. So that's what industry does. And I think me as a government employee, I have a different set of objectives, which is like, I'm not making a product. I'm not trying to compete with the companies. What I'm trying to do is kind of as a backup and a backstop is develop germplasm resources that if there's a need for some of this diversity, either for like a disease resistance or like at some point the breeding pools in the commercial companies just get so narrow and they want to broaden them, that there will be material there that's not going to require them to go all the way back to like an unadapted land race. So it really depends, it depends on who you are and who's paying you for what purpose. So, and that's kind of like my point. Like, I can't really tell you how to breed corn. I'm not a commercial corn breeder. I've never made a product, right? So don't ask me how to do it because I don't know. But what I can do is try to think about how I can do germplasm development in a way that I think might someday be useful to one of the bigger breeding companies.
All right, thank you, Dr. Holland, for that really wonderful presentation. I personally have learned a lot today. So everyone, thank you for joining us for today's webinar. But like I said, this webinar is being recorded and will be posted in the NAPB Graduate Student Working Group YouTube channel. So again, if you want to revisit some points in the presentation or again, rewatch the entire presentation, you could do that. Also, watch out for more announcements from the NAPB Graduate Student Working Group. And I just want to mention that the application for Borlaug Scholarship and George Washington Carver Scholarship is ongoing. So please feel free to apply. With that, thank you and have a good day.
Thanks, y'all.